Now we will be answering different sort of questions in the fields of biotechnology and its applications. Primarily, when you take biotechnology, biotechnology deals with manipulation, experimental manipulation of cells, tissues, organs and molecules such as DNA, RNA and proteins, primarily for genetic improvement, so that we get new traits, and the new traits have to be useful for mankind. So, biotechnology encompasses several fields. We have plant tissue culture, then again we could also be having genetic engineering or recombinant DNA technology. It could also involve microbial biotechnology and even fermentation biotechnology. Industrial microbiology, fermentation biotechnology and microbial biotechnology constitute the first half of biotechnology, that is most of the discoveries which are from say 1920 till about 1980. Then from 1940 onwards till about 1980 we have had very good advances in plant tissue culture. Right from the time we had the discovery of the restriction endonucleases and the human genome project, right from 1980s to 2000, biotechnology has advanced into newer fields like proteomics, genomics and metabolics. However, in the context of MCQs for medical entrance, the questions, the probable type of questions which we are going to exploit out, we are going to exploit a different range of questions which could touch upon changes in chromosome number and structure, tissue culture, biotechnology as well as even transgenic crops. Now we go to question number 1. Question number 1 deals with Tritical. Now, as implied, Tritical is a man-made cereal. So, a Tritical is an artificially constituted plant, and for Tritical is derived through, the options are Polyploidy, that is single plant with increased chromosome number, or B, Haploidy, n number of chromosomes. Polyploidy, when you take for Tritical, you got to cross two different plants whose genetic constitutions are terribly different. And the fourth choice is Polyploidy, that is both self as well as crossing with different plants. Now, as implicated, Tritical is always a relation which has been derived by crossing a sort of a native species with a wild species. Hence, the right answer is Polyploidy. When you say allo refers to two different sort of an organisms or two different crops which have been crossed together, whose chromosome numbers are very, very different, and thereby increase in chromosome number. Hence, the answer is C. The answer is C, and the correct answer relates to Polyploidy. Next, we go to question number 2. Question number 2 says ethyl methane sulfonate, EMS. Primarily, what is EMS? Can it induce polyploidy, haploidy, mutations or protoplast fusion? When you take EMS, ethyl methane sulfonate is an example of a physical mutagen. Is an example of a physical mutagen which can bring about changes in the number, sequence and kind of the nucleotides at the gene level A, and it can also bring about changes in the number, sequence and kind at the chromosomal level. Which can be inherited. And EMS is a correct example or the first valid, well acclaimed example of a physical mutagen. So, the answer is C. While if you take the other choices, polyploidy predominantly introduced by colchicine, haploidy is induced by anther culture, and protoplast fusion is primarily induced by electrofusion. We go to question number 3. Question number 3 asks, what are gene banks? Gene banks is made up of it's a mixture of seed banks, orchards, tissue culture, cryopreservation? Or is it B, seed banks, tissue culture and cryopreservation? And again, the third option is it a seed bank and cryopreservation? And fourth option is tissue culture and cryopreservation. Now, when you take gene banks, gene banks are repositories of the germplasm. They are the repositories of the germplasm which can be preserved in the form of seeds, then it goes to form a seed bank, or it could also be preserved live, that is in vivo, in a botanical garden or a national park. It could also be preserved by tissue culture, wherein you manipulate the cells and the tissues in medium under culture conditions, or else it can also be preserved by cryopreservation. Cryopreservation is low temperature preservation. So, when you take gene banks include in vivo, as well as in vitro techniques. Hence, the right answer for question number 3 is answer is A. Question number 4 relates to genetic diversity. Which one will be searched for obtaining maximum genetic diversity? Recent varieties, old varieties, wild varieties, or all of these? Diversity refers to variety and variability of life forms. In the context of distribution, and conservation of germplasm. When we are looking at genetic diversity, we have species diversity, genetic diversity, ecosystem diversity. When we refer to genetic diversity relates to conservation of the DNA, the genetic material. Conservation of DNA, which can be primarily from recent varieties. You have an improved variety by biotechnology, which could be some of the recent varieties which are adapted to the environment. B, it could also mean old varieties which are preserved by conventional farmers, or old land races which have been preserved. Like if we take the rice germplasm, has been preserved in Orissa. And similarly, if you take oranges have been preserved in Nagpur. Same is the case for apples from Kashmir. Or if you take the wheat variety of Punjab, they are all the older varieties. And sometimes we could also be having wild relatives. Wild relatives of rice are normally present in Andaman and Nicobar Island. So, wild varieties, old varieties and recent varieties, all of these put together constitute genetic variety. You have got to note in the question which one will be searched for obtaining maximum biodiversity. For getting maximum, we require all the three put together. Hence, the correct answer is D. So, while checking on the sort of questions, we have got to read it very carefully, because which one will be searched for obtaining maximum genetic diversity? We are asking about maximum genetic diversity, which includes all the following. Hence, the right answer is D. Next, we go to question number 5. Which one is commonly used for induction of polyploidy? Now, polyploidy primarily refers to polyploidy in crops, refers to increase in chromosome number. Increased chromosome number of polyploidy could include triploids, tetraploids, hexaploids, like you have triploids in the case of watermelons, hexaploids in the case of wheat, and because of polyploidy we have gigas habit. Like larger size of pineapple, larger size of cabbage and cauliflower, and this can be primarily be induced by means of a chemical which is termed as colchicine. Now what does colchicine do? Colchicine primarily causes arresting of the spindle fibers. Arresting of the spindle fibers during cell division, so that chromosomes do not segregate. So when chromosomes do not segregate, we have in the daughter cells an excess number of chromosomes, and the excess number of chromosomes leads to polyploidy. So colchicine induces polyploidy, thereby the right answer is B. While if you take cytokinins promote cell division. Nitrous acid is a sort of a growth hormone. Indolactic acid too is a sort of a growth hormone, and they cannot induce primarily polyploidy because here there is arrestation of the spindle fibers and it interferes with the cell division, especially in the case of mitosis and meiosis in both the somatic cells as well as the germinal cells of plants. Question number 6. Mexican wheat variety introduced in India was. Now primarily when you take plant breeding, in plant breeding, the first step in plant breeding is termed as introduction. Now in introduction, a variety from some other place of not of its origin is introduced into the local area for cultivation. So introduction normally has occurred in the context of wheat in India, which has brought in the Green Revolution. So while we are dealing with introduction, common varieties which have been introduced are the Mexican wheats. And when you take the Mexican wheats, the common Mexican wheats are Sonara 64, as well as you also have Lerma Roja 64A and also Sonara 63. So all these are examples of introduced varieties in our country. Hence the right answer is D. Now question number 7 asks, what is Nonintent gene? Nonintent gene is primarily a dwarfing gene which is present in wheat, and it is initially the sort of a gene which has been introduced in crops. So Nonintent dwarfing with relation to wheat, the right answer is A. Question number 8 relates to amber colored wheat produced from red colored Mexican wheat. Wheat when introduced and bred develops amber colored, and amber colored wheat is seen both in Sharbati Sonara and Pusa Lerma. Hence the right answer is C. It is Sharbati Sonara as well as Pusa Lerma. Now we are going to question number 9. Redley, Sonara 63, Sonara 64 are varieties of wheat. The answer is C. Question 10. Triploids are said to be more useful than polyploids because choice A is they are fruitless and are always propagated by stem cuttings. Choice B is they are produced when definitely nucleus fuses with the male gamete. Third one is they are sterile and show increased vigor. And fourth one is they are fertile and more healthier. When you take triploids, as implicated in triploid watermelons as well as in triploid coffee and tea, they are predominantly they are sterile. They are sterile and they show more vigor. They show more vigor. Hence, the answer is C. Answer is C in the case of triploids. We go to question number 11. Pusa lerma and Sharbati sonara are predominantly mutants of Indian varieties. So, when you take Pusa, Pusa is a very important plant breeding institute which is associated with Indian Agricultural Research Institute at Delhi. It's popularly termed as Pusa Institute, whereby crops are subjected to mutations, mutation breeding. So, among the first varieties which have been developed here, we have Pusa lerma and Sharbati sonara. So, they predominantly are mutants of the Indian varieties. Hence, the answer is B. We go to question number 12. Bread wheat. Bread wheat is popularly termed as hexaploid wheat. Bread wheat is otherwise termed as hexaploid wheat, and the answer is B. Question number 13. Androgenic haploid culture. Androgenic haploid culture or pollen culture or anther culture has been initially studied by Sipra Guha Mukherjee and Pancha Maheshwari of University of Delhi. So, for the 13th answer, always anther culture or haploid culture, especially in terms of rice, or androgenic haploid culture. The right answer among the choices is A. It is Sipra Guha Mukherjee and Maheshwari from the University of Delhi, who are responsible for anther culture and haploid culture, the first anther culture or haploid culture of plants. Next question is question number 14. Improved draft wheat varieties with high percentage of lysine or protein is primarily termed as, it is popularly termed as Sharbati sonara. So, when you take Sharbati sonara, it has high lysine content or high protein content, and the correct answer is D. We go to question number 15. Drafting gene of wheat. Drafting gene of wheat is popularly termed as Noran 10. Answer is C. So, Noran 10, wheat and draft gene. These are the words which have to be associated for the sort of a question. We go to question number 16. When a diploid female plant, that is 2N, is crossed with a tetraploid male, 4N, the ploidy of the endosperm is normally 3N, endosperm cells in the resulting seed is predominantly, it's a triploid, because endosperm is always a sort of a triploid tissue. Hence, the answer is D. Because the endosperm is always triploid, or it is designated as 3N. Next, we go to question number 17. Tritical has been developed through intergeneric hybridization of, the choices are wheat and rye and sea-kale, wheat and agelops, wheat and rice, rice and maize. When you say triticale, triticale is an artificial synthetic, and the artificial synthetic is developed by intergeneric hybridization between wheat and rye or wheat and sea-kale. Hence, the answer is A. It is an intergeneric hybridization. We go to question number 18. A place where endangered genetic materials of the plants are kept is predominantly what it is called as. Choices: A is gene library, B is a gene bank, C is a gene pool, D is a herbarium. Choice number A, gene library. Gene library is an assemblage of recombinant DNA, cDNA, which are present in small petri dishes, which are put in banks. So, a gene bank is an assortment of DNA, the sequences of the DNA and the pieces of the DNA which have got the plasmid. A gene bank, which is an in vitro as well as an in vitro and in vivo conservation of plants, is primarily called a sort of a gene bank. And in a gene bank, we could be having endangered genetic materials like the last resource of any particular plant which has got the sort of a gene sequence for the management or for the cure of cancer or AIDS is normally put in a sort of a gene bank, or the last extinct species which have got high protein content which you could find in a tropical rainforest could also be placed in a sort of a gene bank. A gene pool is the sum total of all the sort of a genetic material of the genera which is present in a natural habitat. We have a primary gene pool of rice and also a secondary gene pool of rice. Similarly, we could also be having a primary gene pool of potato in South America and a secondary gene pool surrounding it. Whereas the last choice, herbarium, is a collection of plant material which is dried, which is primarily intended for A, identification, B, classification, C, comparison, 4, documentation, as well as studying any sort of a relationship. So, a place where if you are dealing with relation to endangered, if you are dealing with endangered plant material, that is which is on the verge of extinction, and it primarily we are dealing with relation to genetic resources, the place where they could be kept is termed as a gene bank. A gene bank is popularly present at International Rice Research Institute at Philippines, and the answer is B. Answer is B. Question number 19 states the first transgenic crop. The first transgenic crop has been with relation to tobacco. We had transgenic tobacco. The answer is B. Question number 20 asks most of the crop plants which are present in the world, they have originated as allopolyploids. So, when you say allopolyploids, that is they have been developed by the crossing of a cultivated species or a natural species with a wild species. A common example is rice. If you take Oryza sativa, Oryza sativa has been developed from both aquatic rice as well as by crossing with a sort of a grass which resembles the rice plant. So, it is an example of an allopolyploidy. Hence, the answer is A. The origin of most crops is by allopolyploidy. Question number 21. New varieties of plants are produced by selection and hybridization, choice A, mutation and selection, introduction and mutation, selection and introduction. We understand that the first step in plant breeding is selection. After you select out, you introduce, then you select. After you select out plants for superior characters, and next you breed them. And the pattern of breeding is or crossing two plants is termed as hybridization. Hence, the new varieties, whenever we want new varieties, the objective is variation. New varieties require genetic variation. Genetic variation, the basis is crossing over. For crossing over, we need to cross two plants. Crossing two plants is termed as hybridization. Prior to hybridization, we need to do selection. Hence, the answer is A. Answer is A, which states that both selection and hybridization are two important prerequisites for the development of new varieties of plants. Question number 22 deals with polyploidy. Polyploidy refers to increase in chromosome sets, and it leads to the rapid formation of a new species. And the reason why we have new species is the choices are: is it due to genetic recombination? Polyploidy does not lead to genetic recombination. Polyploidy does not lead to choice number B, which is isolation. Isolation is when we have a rare line. C, polyploidy does not lead to mutation because there is no change in the number, kind, and the sequence of the nucleotides or at the chromosome level. However, polyploidy results in the development of multiple sets of chromosomes. So, when you have got multiple sets of chromosomes, the multiple sets are the basis for variation. And this results in the formation of new species. Hence, the answer is D. Question number 23 asks, NBPGR is located in New Delhi. What is the full form of NBPGR? It is predominantly National Bureau of Plant Genetic Resources. The National Bureau of Plant Genetic Resources is located in New Delhi, and it deals with germplasm conservation, preservation, and also improvement of the genetic resources, which is the basis for the variability of all the agricultural and horticultural crops in India. Hence, the answer is B. Question number 24 asks, what is hybrid vigor? Hybrid vigor or heterosis is mostly due to, the choices are homozygosity of pure character, B, heterozygosity, C, superiority of all genes, D, mixing up of the cytoplasm of the male with that of the female exclusively. Now, when you take hybrid vigor, we understand in hybrid vigor always the F1 is superior to both the parents or any of the parents. You take an instance wherein you have one of the parents as capital AA, capital BB, small c, and small d. It is crossed with another parent who is small a, small bb, capital C, and capital D. If you take these are the parental lines. Now, in the F1, we have a capital A, capital B from here, and again we have a capital C and a capital D from here. So, A, B, C and D indicates the cluster of dominant genes. Along with the recessive. So, this condition is predominantly termed as heterozygosity. So, heterozygosity results in the F1, especially in the case of hybrid vigor. The answer is B. Hybrid vigor is normally seen in the case of maize or corn. It is seen in maize or corn. Next, we go to question number 25. Question number 25 asks, which is an improved variety of wheat? We understand Sonara 64 is an example of the improved variety of wheat, and the answer is A. The answer is A. Sonara, in the case of wheat variety, is an example of improved varieties. We go on Question number 26. It states Dr. M. S. Swaminathan could produce an amber colored high yielding variety called Sharbati Sonara from Mexican draft variety by using A. Chemical mutagens, B. Radiations of UV and gamma rays, C. Crossbreeding techniques, and D is tissue culture. Development of Sharbati Sonara was somewhere in the 1970s, wherein we could produce a sort of a wheat variety primarily due to mutation breeding, and the right answer seems with relation to mutation breeding using UV rays and gamma rays. Hence, the answer is B. So, Green Revolution in India is related to Sharbati Sonara, which has been primarily developed by mutations and by irradiation genetics. The answer is B. Question number 27 says the wheat variety introduced from Australia is, the correct answer is Ridley wheat. Ridley wheat is an example of introduced wheat, and the answer is A. Question 28 asks, what is Triticum aestivum? Triticum aestivum is an example of hexaploid wheat. Hence, the answer is D. Answer is D. Next question is 29. The indica variety of rice is crossed with the Japanese variety as they are A. high yielding, B. resistant to disease, C. cheaper, D. short life cycle of an annual. Indian varieties have been crossed with Japanese varieties primarily for breeding for resistance to diseases. Hence, the right answer is B. Rice has been developed in India primarily by crossing with the Japanese variety. For the objective has been breeding for disease resistance. We are going on question number 30. Question number 30 relates to tissue culture. Who could grow tomato roots successfully and develop technique of tissue culture for the first time? So, for the first time, tissue culture with relation to roots in tomato has been studied by White. White is related to plant tissue culture. More so with relation to tomatoes. And the answer is B. Next is question number 31. Triticum aestivum, the common breadweed, is a hexaploid. So already we know this. 31, answer is B. Common bread or Triticum aestivum is a hexaploid. So hexaploid weeds consumable. Question number 32. The objective of plant breeding is a science which relates to improvement of crops for better yield, for quality, for disease resistance and stress resistance. Hence, the answer is D, that is all the above. So, plant breeding is a science which deals with crop improvement, especially with relation to improvement of agronomic traits. Plant breeding includes biotechnology. Next, we go to question number 33. What are examples of wheat varieties? Examples of wheat varieties are Roja 64, Sonala 64, Sonalika. Hence, the right answer is D. All of them are examples of wheat varieties. Question number 34. Tritical is obtained by crossing wheat with A. Oat, maize, C. Barley, D. Rye. When we take wheat and cross it with rye, we get Tritical, which is the example of the first man-made cereal. So, among the choices, the right answer is D. We go to question number 35. Noran gene of draftness in wheat has originated through spontaneous mutation at India or Japan, C is Mexico, and D is USSR. Noran gene, primarily wheat draftness gene, has been developed from a Japanese variety. Hence, the answer is B. Next, we go to question number 36. BT or Bacillus thuringiensis has been made through southern blotting, western blotting, cloning, or in situ hybridization. Now, whenever you develop BT cotton, BT Bacillus thuringiensis variety, which are all examples of transgenic crops. In a transgenic crop, there is always in situ or in living state, there is hybridization. That is, we have a vector. The vector carries the foreign gene of interest or the foreign DNA of interest and places it into a bacteria, and it is expressed in the bacteria. Hence, the answer is D. D refers to in situ hybridization. So, in situ hybridization and transgenic crops are related. 37 asks, who is the father of biotechnology in India? The right answer is Professor B.L. Chopra. Answer is A. Whereas Laljit Singh is associated with DNA sequencing, Butler was a mycologist, Maheshwari was an embryologist. Question number 38. Polymerase chain reaction. Polymerase chain reaction was discovered by choices Watson and Crick, McClintock, Carey Millis, as well as a plasmid. Watson and Crick are responsible for elicitation of the double helical model of the DNA. Barbara McClintock is associated with transposable elements or transposable genetic elements or mobile genetic elements. Carey Millis is associated with the polymerase chain reaction. So, Millis and PCR are associated together. Hence, the answer is C. We go to question number 39. PBR322 is a vector. Is it a cosmid? Is it a bacteriophage? Is it a phagemid? Or is it predominantly a plasmid vector? A cosmid is an artificial construct. Choice A. Bacteriophage is a virus which attacks a bacteria. A phagemid is again an artificial insert, but a well-studied vector which is primarily used in genetic engineering is plasmid vector PBR322. P refers to plasmid, BR refers to Bolivar and Rodriguez, and 322 the numerical series of the particular vector, and this is the most efficient vector with relation to genetic engineering, and maybe it is the first generation of vectors which are used in gene cloning. Hence, the right answer is D, plasmid. Next, we go to question number 40. ECOR1, Escherichia coli restriction enzyme R1, is produced from Haemophilus or Morozella or Escherichia coli or Proteus vulgaris. Always ECOR1 is produced from, the right answer is C, that is, it is produced from Escherichia coli. E stands for Escherichia and C, small c, stands for coli. So, it is a restriction enzyme number 1 and it is derived from E. coli. The answer is C. 41 question asks, tag polymerase enzyme is used in A, restriction mapping, B, gene cloning, C, PCR, D, all of the above. When you take tag polymerase, it is a specialized enzyme which has been extracted from a bacteria, and it is a polymerase enzyme which can break or cut the DNA at specific segments. And predominantly tag polymerase is used in restriction mapping, cutting the enzyme, as well as taking the particular enzyme and allowing it to get cloned to a foreign gene of insert. And predominantly tag polymerase helps with relation to both annealing, that is joining, and also it also helps in increasing of the DNA content. So, tag polymerase is a basic enzyme which is used in polymerase chain reaction in PCR. In polymerase chain reaction, a small gamut of the DNA is put and it is amplified. It is amplified by both annealing as well as by renaturation studies. DNA strands are started by a primer. The primers attack the DNA on two sides. There is both separation of the DNA strands as well as joining of the DNA strands, which is termed as renaturation and annealing, and the process is termed as PCR, which has been discovered by Mullis, and the most important enzyme which is used in PCR is tag polymerase. The answer is C. Next question is 42. Which of the following are molecular markers? A is RAPD, B is RFLP, C is SSR, and D is all of these. When you take molecular markers, molecular markers are also called as DNA markers. They exactly know how to take up a particular sort of a DNA and they get themselves amplified. If you take rapid amplification of DNA is by the technique of RAPD. The second method is restriction fragment length polymorphism, that is RFLP, and the third one is termed as single sequence repeats. So, all the following are examples of molecular markers. The answer is D. The 43rd question asks, which is the gas which is used in cryopreservation? In cryopreservation, living material is put in liquid nitrogen, hence the answer is B. Liquid nitrogen primarily at low temperature for preservation of living tissues. We go to question number 44. Germinating barley seeds are used in the manufacture of rum, wine, beer or whiskey. Barley seeds during germination produce alcohols which is used in the manufacture of rum, hence the answer is A. Next, question number 45. Vitamin B12 is obtained from, choice A is Ashby gossypi, B is propionic bacterium, C micrococcus, or Aspergillus niger. It is obtained from propionic bacterium, hence the answer is B. Answer is B, vitamin B12. Question number 46. Gibberellic acid, a growth hormone, is obtained from, it is predominantly obtained from a fungus which is termed as Fusarium monoliforme. Hence the answer is C. Answer C, Fusarium is a source of gibberellic acid. Question number 47. Gene sequencing is used for determination of the base sequence of DNA, RNA, proteins, or all of these. Sequencing of genes refers to, when you say gene, gene is a stretch of nucleic acid. It is predominantly made up of nitrogen bases, and gene sequencing refers to the master molecule, and the master molecule is always the DNA. Hence, gene sequencing is associated with the base sequence of the DNA. The answer is A. Question number 48 asks, which of the following is a method of gene transfer? Whenever we have a foreign gene of interest, the foreign gene of interest can also be transferred by physical methods. The physical methods could involve A, micro injection with a needle, B, a particle gun. In a particle gun, we have the gene of interest which is just put in and shot into the sort of plant tissues with a radioactive compound. C is by electroporation. Electroporation is when you use electrical discharges and you allow a sort of foreign gene of interest to get into the protoplast. So, all A, B and C combined together are methods of gene transfer. Hence, the answer is D. Question number 49. Which of the following techniques is used for recovering of interspecific hybrids? When you take interspecific hybrids, are predominantly recovered by means of a technique which is termed as embryo culture. When you take embryo culture, when you culture embryos, you can rescue embryos. You can rescue embryos. In embryo culture, you can recover or rescue interspecific hybrids. Interspecific hybrids. Hence, the answer is C. We go to question number 50. Haploid plants are obtained from either anther culture or meristem culture or embryo culture or all the above. Normally, when you take haploids, haploids are developed by anther culture. Because when you take the microsporangium is haploid. Hence, the answer is A. Answer is A.